Now look at this.
What's up, Big Penguins? Today we're going to do topic 2.10 on compartmentalization. So I think we can all agree that eukaryotic cells are going to be cells that have membrane-bound organelles. So most of the organelles that we see in the cell, the lysosome, the ER, the Golgi, the peroxisome, the nucleus, all of them are surrounded by a membrane. And this membrane allows us to have these multiple different compartments within the cell. And by having these compartments, we can have specialized reactions taking place in each of those compartments. Due to that compartmentalization, that's why the eukaryotic cell is able to be so much bigger or larger than the prokaryotic cell. Okay, y'all. So this diagram is a little bit more complicated than it needed to be, and it's not really showing what I need it to be. So just bear with me for just a second, okay? So this is the mitochondria. In the mitochondria, this is the site of oxidative phosphorylation, and this is where we're going to synthesize ATP. There's an inner membrane called the cristae. And so within the cristae is this place called the matrix. That's where we're going to see the Krebs cycle taking place. And then along the cristae is going to be the site of oxidative phosphorylation. That's where we have the electron transport chain that's going to pump protons from the matrix out into the IM space. So in this picture, you see the inner membrane, the cristae, and then the outer membrane. So this space right here is where we're going to pump those protons out to. So we have a compartment that we're going to have those protons in that then allows us to have the protons flow down their concentration gradient back into the matrix, which is what's going to synthesize the ATP. So as we see, compartmentalization is allowing us to do this process within the mitochondria. So the ER is going to have the function for, of course, compartmentalization, right? So we look at our smooth ER. Smooth ER is going to be a storage location for calcium, so we can store calcium in it. And calcium is a secondary messenger. And so due to that compartment, we're able to store calcium in a region of our cell to be used later on in another process. In our rough ER, you're going to see that we have these ribosomes studding the membrane. And so the large surface area is allowing us to synthesize proteins inside of this membrane, right? In this compartment. To secrete those proteins. Fun fact, all ribosomes are cytosolic ribosomes until they get the signal to move to the membrane. So here we see our chloroplast. Chloroplast is the site of photosynthesis. Inside the chloroplast, you have these little stacks called grana, and those stacks are made up of thylakoids. So the membrane is the thylakoid membrane. And so you see that we have this little photosystem in that membrane. And the same thing we saw in the mitochondria. There's going to be an electron transport chain that's going to pump protons. It pumps those protons into the thylakoid space, so inside of that little sac. And so again, we have a compartment to hold those different ions to allow us to generate that ATP. The Golgi is made up of multiple sacs, and these sacs allow us to sort and modify the different products from that rough ER as we move through the different sacs. If you have pains or just success, and don't forget to follow for more.